Good day, class, and thank you again for tuning on Metric Hub. We are still going through our revision and preparation for end of year exams, picking and touching every area that we think is necessary. Today, I'm just going to bring to you this meiosis, specifically checking on what is present on the body, the consequences of abnormal meiosis. Abnormal meiosis is when meiosis does not take place properly, or there are errors, there are disorders that arises during meiosis, which we are saying that we need to look at the end results, the effects, if meiosis does not occur properly. And I always want you to understand that the primary importance of meiosis is the formation of gametes. When gametes are formed during the process of what? By the process of meiosis. Meiosis enables sex cells to be formed. So obviously, you should understand that meiosis, it is primarily important for halving the number of chromosomes that are found in a gamete. So if ever there are abnormal stages or abnormal areas, abnormal effects that might arise, therefore it means then that the gametes are affected. And then if we don't have gametes, we don't have seeds. Seeds are very important. Seeds enable us to have succession. Seeds promote development and continuous establishment of a species or of a of a given population. So non-disjunction of chromosomes is actually a typical example of abnormal meiosis, non-separation. We call that non-disjunction or non-separation because you should know that these words are the same. Non-disjunction or non-separation of chromosomes. So sometimes the following mistakes may occur during the process of meiosis. So during anaphase 1, one or more homologous pairs of chromosomes may not separate. One or more homologous pairs of chromosomes may not separate, and understand that it is anaphase 1. It occurs in anaphase 1. So during anaphase 2, the sister chromatids of one or more chromosomes may not separate again. This is known as what? Non-disjunction, which I also said that is also called what? Non-separation. I said is also called non-separation. As a result of that non-separation, gametes forming from meiosis may have one extra chromosome. Gametes forming from this meiosis as a result of non-separation may have one extra chromosome, or it can also have one less chromosome, no chromosomes, or an extra set of chromosomes. All these are effects, or these are possibilities that we can have. So having said that, we have said, you need to understand that such an abnormal gamete may fuse with a normal gamete. Whatever gamete that is formed from non-separation or that has been formed where non-disjunction has occurred, it can fuse or it can fertilize or it can join with a normal gamete or another abnormal gamete, leading to different genetic disorders. I'm sure that you know genetic disorders, we are talking about diseases or conditions that are inherited, which we also call congenital conditions, which we are saying they are passed because of the lineage, they are passed on from generation to generation. They can be inherited as a result of the what? Of the genes that are carried by the chromosomes, which I'm sure that we strongly explained and dwelled more on that. So non-disjunction may lead to two general conditions which can be called aneuploid or polyploid. There are two conditions that can result from non-separation, which can be aneuploid or polyploid. Let's look into aneuploid. In aneuploid, one gamete receives two copies of the same chromosome. In this condition, we are saying one gamete receives two copies of the same chromosome, while the other gamete receives no copy. It means it has nothing. So if fertilization occurs with either of these gametes by a normal gamete, the zygote will have an abnormal number of chromosomes. That is either one less or one more extra chromosome. In this regard, again, if the gamete has one extra chromosome, we say it is trisomic for for that chromosome. We are saying if the zygote is one extra chromosome, it is termed as trisomic for that what chromosome, as to say that it has got extra, it has got three copies of the same chromosome. If the gamete is one chromosome short, it is called what? Monosomic for that chromosome. If it is short, class, if it is short, it is called monosomic. And then Down syndrome is an example of the result of aneuploidy. This has been dealt with in more detail, as you can see below here. We are going to talk about that. It is a result of extra chromosome number 21, Down syndrome, a condition that is resulting from non-separation of chromosome, which is we are referring to on this in this regard is trisomy condition, which is caused because of chromosome 21. Since the cells are trisomic for chromosome 21, Down syndrome is also called trisomy 21. It can also be called trisomy 21 Down syndrome class. And then in meiosis 1, the chromosome pair 21 may not separate, or in meiosis 2, the chromatides of the chromosome 21 may not separate again. Take note of the situation here. We are saying in meiosis 1, the chromosome 21 pair may not separate. When it is meiosis 2, it is the chromatides that may not separate also of chromosome 21. And some gametes will therefore have an extra copy, that is to say 2 copies of chromosome 21. So if it is having 2 copies of chromosome 21 and then it fuses with a normal gamete, therefore some gametes would therefore have an extra copy of 2 copies, or 2 copies of chromosome 21. If a gamete with 2 copies of chromosome 21 actually uh, fertilizes or fuses with a normal gamete with 1 copy of chromosome 21, the resulting gamete will have 3 copies of chromosome 21, because what we are saying is that if the gamete with 2 copies, it has got 2 copies of chromosome 21, and then it fuses with a normal gamete with 1 copy of chromosome 21. Therefore, the resulting gamete will have 3 copies. It will have 3 copies of chromosome 21, or we can say that 47 chromosomes instead of 46. That is resulting in what? In chromosome, it is resulting in what we call Down syndrome. I'm sure this we are making sense and we can easily move fast and understand this. Individuals with Down syndrome are characterized by mental retardation. Sometimes, of course, not many times anyway, but I'm saying there is questions that sometimes come there where you need to give characteristics of Down syndrome children or Down syndrome individuals. So people who are Down syndrome, they've got problems or they are characterized by mental retardation, as to say their mental alertness and judgment is not does not grow fast, does not link to their age and so forth. Their reasoning is not quite at par. So they also suffer from hearing loss. They also have problems with the heart defect, decreased muscle tone, upward slanting eyes, a small mouth, which is also accompanied with a small nose. There is abnormal ear shape and a depressed nasal bridge. A depressed, as to see, they don't have a nasal ridge. The bridge is not there, it is depressed. And there is no cure for Down syndrome anyway. There is no cure for that. Only the symptoms are treated. The symptoms can be managed. To detect the occurrence of Down syndrome in unborn babies, fertile cells are removed from the uterus of the pregnant mother by amniocentesis and analyzed. I always have got a soft spot for this process or this word, amniocentesis. It is the collection of amniotic fluid from pregnant mothers for analysis and detection of congenital disorders or diseases. So as you know that when the baby is growing inside the mother's womb, the cells are always dying and being replaced. So the amniotic fluid that surrounds the baby that we always talk about when we are talking about the safety growth of the child, which we talk in regard to avoiding of drying out of the child, the flexibility of the child, the absorption of any other pressures and physical mechanical forces. It is because of the amniotic fluid. So the amniotic fluid, it would also contain the cells or the foetal cells, which can therefore be collected, that fluid, for what? For amniocentesis. So amniocentesis, ne, it actually helps in identification, or it helps in monitoring of the monitoring of welfare and growth of the child before birth, when is the baby, or during pregnancy. So therefore, congenital problems can be identified, problems that are any disorders or disabilities that might be affecting the child can therefore be picked because of amniocentesis. So please take note of that. And then the karyotype resulting from such analysis is then examined uh, to determine if there is an abnormal number of chromosomes. The abnormal number of chromosomes is one thing that you need to understand. So with this in mind, I want you to know that karyotype, what is it? It is the structure. Karyotype is the structure. It is the structure and arrangement. It's the structure and arrangement of a person, of an individual chromosomes. So the structure and arrangement of a person's what chromosomes is, we call the what, the structure and arrangement. It is called what? It is called the karyotype. So in terms of the karyotype, you must understand that we have got 23 pairs of chromosomes, which we know that from number 1 to number 22, they are all autosomes. And then we are saying 23 is the sex chromosome. Chromosome 23 is the sex chromosome. Is the sex chromosome. This one is the sex chromosome, chromosome 23. They also gonna call it gonosome and other words. So with this in mind, we want to dwell more on the issues of what, of trisomy condition. So what happens is that when we are given the karyotype, there are questions that comes as to say, identify. First question, which is there, is to, I say, is this a karyotype of a normal or abnormal person? So we are saying when there is three pairs of chromosome on chromosome 21, we call that what is called, it's an abnormal. This is an abnormal. This is called an abnormal karyotype, or the person is abnormal. Because we have got what? Because we have got trisomy, we have got trisomy 21. Trisomy 21 is given there, which you can see there is trisomy 21 here. So having said that, the next question is that give the reason, or what causes this trisomy 21. What causes trisomy 21? The process that causes this is non-separation of chromosome. Trisomy 21 is caused as a result of what? Of non-separation of chromosomes, where we defined, which we explained up there, that in meiosis 1, chromosome 21 did not separate, and then it fertilized with a normal, it actually fertilized with what? with a normal gamete, which resulted in what? In which resulted in two copies of chromosome 21 fertilizing with one with a gamete with one copy, resulting in trisomy 21 that you see there. And then we are saying it is caused because of non-separation of chromosome. And then an individual who is having this condition, we call that the person is what? Is got Down syndrome, or we are saying is a Down syndrome individual. And then I talked about the characteristics there that I've explained there. So another question again in terms of the karyotype is a question which has to do with the gender of the person, the gender of the individual. Where there is a question to do with what? With the gender in terms of what? In terms of the karyotype. So for that, you should know that you consider chromosome 20, chromosome 23, chromosome 23, if it is a male, you should know that we have got a what? A long chromosome and also a short chromosome. So this represents a male person, a male individual, because of the presence of a what? Presence of a short Y chromosome that you can see here. I'm saying you can see that we have got a what? We have got a short chromosome. This one, it is short. Chromosome Y is short there, which I'm saying this is X. This is X, and this one is what? This one is Y. As you can see there, we have got an X and Y chromosome, which is shown by a short what? By it being short like that. So please note that this is a what? Gender of a male. And then we can as well say that the one which is what? The gender, we are saying a female is like the one that is shown here, which is shown by what? By double X chromosome, which is indicated by what? Uh, which is indicated by same length of chromosome, same long Y chromosomes. Both of them are long like that. That is representing to say that this is a female. It is a female because we have got what? We have got both long X chromosome, both long X, both long X chromosomes represent that the person whom we are referring to is a what? Is a she. This is a female because of the double X chromosome. XY is shown by having what? A short Y chromosome that is there. So I'm sure that you can see this, and thank you so much for following, and all the best. Keep on connected until the year is done, and know that you never walk alone when Metric App is available for you. All the best. You have a beautiful study, uh, and just watch forward for any other coming, for plenty of upcoming videos with regard to all papers. Thank you.